Hello, my name is Kevin Tolalpa, and my project focuses on studying the potential interaction of the microRNA MIR25-3P with the polyphonic compound called epigallocatechin 3 gallate. Our work wants to better understand what happens in breast cancer, the most common cancer among women worldwide. About 1 in 8 women will be diagnosed with breast cancer in their lifetime, and 1 in 39 will die because of it. A population study in Japan has shown that the consumption of green tea improves the prognosis of breast cancer, and this was attributed to the presence of polyphenolic compounds called catechins in this beverage. An example of a catechin compound is epigallocatechin 3 gallate, and here we can see a structure of it. These compounds have been found to modulate the expression levels of microRNAs such as MIR25-3P. Studies have shown that epigallocatechin 3 gallate downregulates the expression of microRNA 25-3P in breast cancer cells and multiple myeloma. The goal of this research was to predict the secondary and tertiary structures of microRNA 25-3P and determine if they contain cavities that can be potential spaces of interactions with small compounds such as epigallocatechin 3 gallate. For this experiment, different bioinformatic programs were used to predict the structures of MIR25-3P. To start, we retrieved the sequence of MIR25-3P from MIR base. With the RNA sequence, we used RNA structure web, RNA fold to predict the 2D structures of MIR25-3P. iFold was used to generate the 3D structures and to predict clusters in MIR25-3P, and visual molecular dynamics allowed us to turn the clusters into 3D images to help us identify for the presence of cavities. In Figure 3, in the results, we show the predictions of 2D MIR25-3P structures. The different color on the bases represent different base pair probabilities. The red represents a 99% base pair probability, and the light pink represents a 50% base pair probability. We see that MIR25-3P can also form hairpin structures. In Figure 4, these are different structural representations of MIR25-3P on visual molecular dynamics. Figure A shows a CPK space-filling model, and Figure B shows a licorice representation. Figures C and D are molecular surface representations in which we indicated in yellow the presence of a cavity. We consider this a cavity because it has an opening on both sides of the microRNA. These results suggest that in the structure of MIR25-3P, there is a potential location where epigallocatechin 3 gallate could bind and in this way interfere with the expression. In further studies, we will use a bioinformatic prediction approach to understand how epigallocatechin 3 gallate and other catechins might bind to the predicted cavity on MIR25-3P. Thank you.